Hello, everyone. Thank you, Megan, for the introduction. I would like to now take this opportunity to discuss a case study where we use modeling and simulation to enhance our understanding of structure-performance relationships for Dapsone topical gels. The objectives, learning objectives for today, I will discuss in silico methodologies for topical products that are applied on the skin. I will explain the data requirements and scientific considerations when building and validating dermal physiologically based pharmacokinetic, PBPK in short, models, in this case for Dapsone topical gels. And we will recognize how in silico models can be used to support regulatory decision making. The outline of the talk, Dr. Kelchen did an excellent job describing what is the rationale, the thinking behind developing product-specific guidances, what are the considerations. I will just highlight some relevant ones, relevant to the specific example I'm discussing. And then I will give a little bit of information on what are in silico methodologies for topical products. And I will spend most of the time talking about the case study, a dermal PBPK model that was developed for Dapsone topical gel, 7.5 and 5%. What were the considerations that went into the development, validation, and the application of the model? So PSGs for topical products contain harmonized recommendations that are in alignment with relevant general guidances, such as the Q3 characterization guidance, the IVRT studies for topical drug products guidance, and the IVPT studies for topical drug products guidance, all submitted for ANDAs. When the recommendations, when the PHGs contain recommendations within a characterization based by equivalence BE approach, then these BE recommendations are product specific and they're based on an understanding of the microstructure and performance of the drug product. I believe that Dr. Kelchen made that clear. However, BE recommendations in the product-specific guidances may also include in vivo BE studies with PK endpoints or comparative clinical endpoint studies. So this talk will focus on a revision, PSG revision, that took place in February 2024. And this PSG revision refers to two products, the Dapsone topical gel 7.5% and the Dapsone topical gel 5%. Prior to February 2024, the PSG contained two options for this product. So for test products that met the no significant difference criterion, what was recommended was an in vitro characterization approach that required Q3 sameness and a comparative IVRT and IVPT passing studies. And this approach was supplemented by an in vivo BE study with PK endpoints. The option 2 was reserved for test products that did not meet the no significant difference criterion. After February 2024, this guidance was revised, and option 1 does not include anymore an IVPT study or an in vivo BE study with PK endpoints. The rest of the in vitro characterization studies under option 1 were maintained. This PSG revision, as Dr. Kelchen pointed out, was the result of our improved understanding on the product structure, microstructure, but it was also supported by modeling and simulation. This approach, the modeling and simulation approach, elucidated the relationship between drug product quality attributes, which are the Q3 characteristics that are included in our PSGs, and the in vivo performance of the product. And these models that I will go into a lot more detail next establish the link between systemic and local Dapsone exposure. So very, very quickly, the in silico methodologies that I will be discussing today are essentially mathematical statistical models that describe how the amount of Dapsone in this case changes with time based on an administered dose. To generate this output, these models take into account information on the in vitro performance of a product. This could be, for instance, an IVPT study in the case of the Dapsone gel. Information on the physiology, the skin, what are the physiological parameters that impact permeation through the skin and distribution throughout the entire human body. And also, these methodologies account for drug product characterization information. So a particle size distribution would be an example of this type of information. Now, for this case, we developed two models for these Dapsone gels, different strengths, 7.5 and 5%. The objectives of these models were to understand the factors that are governing the permeation of Dapsone through the skin, identify which drug product quality attributes that are specific to the Dapsone topical gels may impact local and systemic bioavailability. And these models were finally applied to perform virtual bioequivalence assessments that evaluate the impact of formulation differences on BE outcomes. These virtual bioequivalence assessments were performed at the level of the plasma, so systemic exposure, following the application of the gels, and also within the context of an IVPT study, an in silico IVPT study. In order to develop these models, we leverage data on the in vivo permeation, the in vivo absorption of Dapsone gels, and that is described through the model, schematic on the top here. But also, at the same time, we leveraged information from IVPT studies by developing an in silico IVPT model that really captured all the experimental conditions that are relevant to an IVPT study. These models work together to develop and validate each other and result in reliable, realistic predictions of Dapsone permeation through the skin and absorption into the systemic circulation. So this is the general workflow that we followed. It is very similar between the topical gel, the 7.5 and the 5% strength. So we started with developing a systemic disposition model for Dapsone. We leveraged literature data to do that. And then we started focusing more on the skin absorption component of that model and developed the Dermal PBPK model for the 7.5% gel. We used data from approved ANDAs to optimize certain components of the model. And that optimized model then was validated against additional independent data sets that were extracted from other approved, additional approved ANDAs. Once this model was validated, we applied it towards virtual bioequivalence assessment for all ANDAs that were considered for development and validation. And in addition to the plasma BE assessment, we also were able to perform bioequivalence assessment in the stratum corneum and the dermis for this product. What was also unique in this specific case is that we were able to validate the model using IVPT data. And in my next slide, I will go into a lot more detail on this. The workflow for the development and validation of the Dapsone topical gel 5% model is very similar. We started with the same systemic disposition model. And now we switched to ANDAs, approved ANDAs for the Dapsone topical gel 5%. We saw that the model for the 7.5% topical gel performed really well. So all the data from the approved ANDAs for the 5% was considered external validation. We did not need to optimize the model at all for the 5% strength. And similarly to what we did earlier with the higher strength, in this case, we applied the model for VBE assessments for the ANDAs, approved ANDAs for the 5% strength in the stratum corneum and dermis, so locally and systemically. Now, we just discussed the workflow for the 7.5% gel, and I said that we used IVPT data to further validate this model. So in parallel to this specific in vivo model, we developed an in silico IVPT model, which described the IVPT study conducted for the Dapsone topical gel 7.5% within an approved ANDA. We optimized certain parameters of the model. I will describe those in my next slide. And we used that partially validated IVPT model, in silico IVPT model, to assess bioequivalence between the RS and the test product in the skin, the sternum corneum, and also the receptor media. So this is a schematic that will help you understand how these models are built. They're essentially mathematical equations that describe how the API is partitioning, is being released from the formulation, which is the gray compartment here, into the sternum corneum and the rest of the skin layers. In order to describe mathematically this process, we can account for Q3 aspects of the product, such as formulation pH, particle size distribution, apparent viscosity, and solubility. So these are direct inputs into the model, and there's information that is collected within the scope of the Q3 characterization of the product. Nothing really is required in addition to those Q3 studies. We do account for formulation composition when we are building these models. As I mentioned, the Q3 aspects, the drug product quality information is included into the model. And for validation, we used in this case in vivo systemic PK data and IVPT study data. We made certain assumptions regarding the evaporation and drying rate of the gels, but this can also be characterized experimentally. And in the case of the in vivo application, we did not assume any rubbing, although this was reported in the clinical study protocols. So we utilized these models for VB assessments. We performed 10 virtual BE trials where we matched essentially the in vivo BE studies that were conducted in support of the approved ANDAs we worked with. And we assessed bioequivalence virtually for Cmax and AUC in the sternum corneum, the dermis, and the plasma. In terms of the IVPT studies, we again matched the conditions of the IVPT study in terms of donor demographics, replicates, and dosing conditions, and the experimental conditions. And we assessed B for maximum amount and AUC in the sternum corneum for Dapsone and cumulative amount and Jmax of Dapsone in the receptor solution. This slide summarizes the performance of the model, where we see that overall the blue lines that are the model predictions overlay relatively well with the observed data, which is the black dots. Now, when it comes to IVPT data, we developed an IVPT model, as I mentioned earlier, and in this case, what we see, the model predictions are the red and blue lines, and the observed data are the squares. What was interesting here, and I would like to note, is that what we saw is that when we include variability in the applied formulation on the receptor, on the donor side, we were able to describe the variability that we see in these type of studies very well. So that's an interesting point to consider in the future. Similarly, in the case of the 5% gel, we're summarizing here the performance of the model, predictions and observed data overlay relatively well. So these models were applied to assess bioequivalence between the RS and the test product in the sternum corneum, the dermis, and the plasma across all the ANDAs that we utilize for model development and validation. And we saw that when we have a test product that meets the no significant difference criterion, Q3 sameness between those two products leads to BE locally and systemically. And certain Q3 attributes, in this case particle size distribution and apparent viscosity, impact BE outcomes and should be closely controlled. Same were the outcomes in the case of the virtual IVPT studies, where we looked at Jmax and the cumulative amount in the receptor solution and also amount of Dapsone in the stratum corneum. All of these ANDAs were considered were found to be bioequivalent, and similarly to the in vivo study, this modeling and simulation approach showed that Q3 sameness between the reference and the test leads to BE locally and the receptor solution. So in conclusion, this modeling approach improved our understanding of the structure-performance relationship of Dapsone gels. Apparent viscosity and particle size were shown to be important Q3 quality attributes. And these developed models can serve as tools supporting a risk-based assessment. The compelling evidence from this PBPK modeling approach aligns well and reinforces the BE recommendations in the aforementioned draft PSGs for other topical gels, as Dr. Kelchen pointed out. So first challenge question: which Q3 attributes were identified as impacting bioavailability based on the developed dermal PBPK model? Formulation pH, rheological properties, particle size distribution, and droplet size distribution. And the answer is B and C. Challenge question number 2. Enhanced understanding of the structure-performance relationship of Dapsone gels gained through the PBPK modeling research led to a successful product development meeting for an applicant, an ANDA approval, or a PSG revision. And the answer is a PSG revision. Thank you very much for your attention. I would like to thank everyone, my colleagues at DTP1 and DQMM, for their support on this project. And we can move on to the next talk that will be delivered by Dr. Tanaz Ramizanli on the approaches for evaluation of formulation differences on performance of topical products. Thank you.